Moving on to specification number 3, which states, state and apply the formula, magnification equals image size divided by the actual size. Understanding this concept is crucial in fields like microscopy, where magnification is used to reveal finer details of small objects or specimens. The key concept of magnification to remember is that it describes the size of an image in comparison to the actual size of the specimen, essentially showing how much larger the image appears relative to the specimen's true dimensions. To calculate magnification, you need to use this formula, that is, magnification equals image size divided by actual size of the specimen. This formula helps determine the magnification factor, a numerical value that tells you how many times larger the image is than the specimen itself. Take a look at this triangle diagram, also known as the magnification triangle. It's a simple yet effective tool that helps students understand and apply the magnification formula in microscopy. The triangle has 3 corners, each representing a key variable, I for image size, which is the size of the object as seen through a magnifying instrument, M for magnification, which is the factor by which the image is enlarged relative to the actual size of the object, and A for actual size, the true size of the object. By using this diagram, students can easily rearrange the formula to solve for any of the 3 variables. For instance, to calculate the image size, cover the I and the formula becomes image size equals magnification times actual size. To find the actual size, cover the A and use actual size equals image size divided by magnification. Finally, to determine the magnification, cover the M and the formula simplifies to magnification equals image size divided by actual size. This visual representation makes it easier to understand the relationship between these variables and apply the formula in various contexts. Also, in microscopy, 3 key terms to remember are magnification, scale bar, and units of measurement. Just know that the magnification refers to the ratio of the image size to the actual size of an object, indicating how much larger the image appears compared to its true dimensions. A scale bar is a short line placed on an image or micrograph that represents the actual size of the object, providing a visual reference for its true scale. Common units of measurement in microscopy include micrometers, which are ideal for measuring small objects under a microscope, and millimeters, typically used for larger measurements in practical applications. Understanding magnification and scale bars is vital for accurately interpreting images, as it allows one to determine both the degree of magnification and the actual size of the objects being studied. The units of measurement are important because accurate calculations are needed to figure out the magnification of tiny structures, which are measured in very small units. In biology, especially when studying tiny things like DNA, viruses, bacteria, and human cells, understanding these units of measurement is important. For example, DNA is the smallest structure, measuring around 10 nanometers, and needs high magnification to be seen. If we magnify it 10 times more than the level needed to view DNA, we can see a virus, which is still small at about 100 nanometers but larger than DNA. With 100 times more magnification, a bacterium, around 1000 nanometer in size, becomes visible. Further magnification, up to 1000 times, allows us to see human cells, which range from 10,000 to 100,000 nanometers. Knowing these measurements is key for accurately seeing and comparing the sizes of these microscopic structures. To measure objects at different sizes, scientists use various units: the nanometer for tiny things like DNA and viruses, the micrometer for cells and organelles, the millimeter for larger objects like insects, and the centimeter for everyday items. A scale bar on images helps show the relative size of these objects, with 1 centimeter equal to 10 millimeters, 1 millimeter equal to 1000 micrometers, and 1 micrometer equal to 1000 nanometer. This scale makes it easier to compare the sizes of biological objects, from the very small DNA and viruses to larger bacteria and human cells. This visual comparison helps to illustrate the vast range of object sizes, from the microscopic to the macroscopic. A horizontal scale is used, with measurements in micrometers increasing from left to right. Objects are placed along the scale based on their approximate size, and the scale is divided into sections, each labeled with the type of microscope needed to observe objects within that range. On the microscopic end, we start with very small entities like water and sugar molecules, followed by larger biological structures such as antibodies, viruses, and bacteria, all of which require a microscope to be seen. As we move along the scale, we encounter amoebas, single-celled organisms visible with a light microscope. On the macroscopic side, objects like a period, full stop, or an orange are large enough to be seen with the naked eye. This comparison highlights the significant size differences in nature, emphasizing the limitations of the human eye in detecting the smallest biological structures and the need for microscopes to study them. The scale bar provides a helpful reference for comparing the sizes of these objects relative to each other. But what we need to learn are these conversions, which we'll also practice in the upcoming examples. Just remember that a micrometer is 1/1,000,000th of a meter. To put this into context, 1 meter equals 1 million micrometers, 1 centimeter equals 10,000 micrometers, and 1 millimeter equals 1,000 micrometers. Micrometers are the standard unit of measurement in microscopy because they are ideal for measuring very small objects, such as cells or the width of a hair. Overall, understanding these conversions allows us to better understand the scale of microscopic objects in relation to those we can see with the naked eye.